Sarah Atzet from Sentrx Animal Care, and I'm excited to be talking to you today about an introduction to our BioHance cross-linked HA. This is me and my dog Dottie. She's a sheepadoodle, and she's a great companion and also has been helpful here at Sentrx. You'll see a couple pictures of her later on, even in the presentation. But my background is in biomaterial development, chemical engineering specifically, and I've been in the animal health field for about 10 years and working at Sentrx in their manufacturing operations and research and development. Sentrx is a Salt Lake City-based animal health company, so our products are based on this patented and proprietary BioHance technology. And really, as a company, we are focused on easy-to-use care solutions that promote the long-term well-being of animals. We do all of our manufacturing and development here in-house and have products both in ophthalmic and wound care for companion animals. And even though Sentrx is located here in Salt Lake City, Utah, we do have a global footprint and have sold millions of doses of our technology to veterinarians around the world. And we're really excited to be adding Brazil with the help of DrogaVET to this footprint. So I thought I'd spend a minute talking about what is hyaluronic acid, and some of you may be more familiar, but I thought I always love to talk about it, so here we go. It's often described as a goo molecule. It's a long linear polysaccharide and a major constituent of the extracellular matrix. It's really negatively charged, and so that anionic property attracts water, and then the body uses it as a lubricant in many different ways. But it's found in neural tissue, epithelial tissue, connective tissues, and it's predominant in the eye as well. Historically, products were made from HA that had been isolated from rooster combs, but almost now all of it is synthesized from bacterial fermentation. And it has some really interesting physical properties. It's viscoelastic in nature and also shear thinning. So as you apply pressure, then the viscosity of the material decreases. It's used in many different medical products, including eye drops, intra-articular joint injections. It's dermal fillers, and wound healing products are all on the market that are based on this HA. And you'll also find it in many beauty products and cosmetic lotions. So what is our BioHance technology? BioHance is what we founded our cross-linked HA. And cross-linked versus linear HA, I'd like to delve into that a little bit. It's found in the extracellular matrix as a linear structure, so that's how it's synthesized and found in the body. But it can be cross-linked to form a hydrogel, and this hydrogel is just a three-dimensional network of covalently bonded polymers, which increases the amount of water that it can really hold. And the style of cross-linking that we perform allows us to have a really purified end product, and so we have no residual cross-linker, which in other cross-linked HAs can be what is often irritating or can cause inflammation. And by cross-linking it, you get a number of different advantages. You can extend the product contact time on the ocular surface from 15 minutes to over 2 hours. And it really just prevents the product from degrading as quickly and maintains a protective barrier and can really help promote wound healing. So this is the actual chemistry that our products are based on. You can see hyaluronic acid in its native form here. And then we chemically modify a few groups on its backbone and cross-link them together to form this polymer mesh, this polymer network. And you can see here there are some really interesting physical properties. So the viscosity profile is a really great way to measure how a polymer behaves physically, and shear thinning is the property when you apply pressure as you would in a blink, and when that happens, then the viscosity drops and the solution flows easily. And when that pressure is removed, then the viscosity increases again. And cross-linked HA has really even more shear thinning than a linear HA. So you can see here, especially at lower shear, compared with HA, you can see that decrease in viscosity. So it makes a really great eye product because it can be redistributed and minimize blurring on the eye. These are Sentrx's BioHance products that are on the market in the US, and I guess these lower images are some that may be found in other global areas, but these are the ones that we predominantly sell here in the United States, and we'll be looking forward to working with DrogaVET to get some Brazilian products similar to these on the market. So this Bioenhance technology has really been scientifically studied and really well evaluated through many different peer-reviewed publications, starting really with the beginning publications describing the chemistry, and then looking at a number of different applications in wound healing, in tendon healing, and then looking more closely to corneal abrasions and ophthalmic uses. And I just wanted to pull out a couple of highlights from some of these peer-reviewed studies. So in 2017 in the journal Cornea, they reported that corneal ulcers in dogs and cats were treated with either cross-linked HA or linear HA, but those treated with cross-linked HA healed significantly faster in both species than compared with linear HA, and certainly even more so than compared with untreated or control groups. And then in 2014, in a study looking at KCS or dogs with dry eye, this was a mass study when they were treating with cross-linked HA versus, again, a tear supplement that contained a linear HA, and they were able to see a significant improvement in ocular health here. So in conjunctival hyperemia, ocular irritation, and ocular discharge. And in addition to that, that last study also looked at pet owner happiness, and you can see that in this mass study that pet owners felt like they were happier with the Bioenhance Ocunovis results, and that it was more effective, especially when compared to the linear HA teardrop that they were using as a control. I just wanted to share a couple of case studies with you that we've had reported to us over the course of our product history, but this is a really unfortunate event where this poor cat Luigi got a thorn after a late night adventure that was stuck, you can see, pretty deeply into his eye there. And 28 days after his first veterinarian visit, and after applying our Oculenus Bioenhance technology, you can see that they maintained vision and had a really great result. And then here are some more examples of the dermal, the wound healing application, and you can see that wounds that are really complex are healing quite nicely and quickly, and you have really improved healing in a lot of them. So I want to talk today, though, about kind of two current ophthalmic indications. The first one is the extended lubrication and comfort for dry eye and KCS application. And we just had a talk this year at ACVO by DJ Heusler talking about this extended residence time and the impact of that on lubrication. So the objective of the study was to compare the residence time of cross-linked versus linear HA on the ocular surface of dogs. And in order to do that, we tried to hold all other parameters constant, and we compared our Oculenus, which is a 0.75 cross-linked HA, we sometimes abbreviate as XHA for short, versus a linear HA. And we chose a linear HA that had a similar molecular weight to other products that are on the market. So its molecular weight was approximately 1 megadalton. And then we also used saline containing fluorescein as a control. It was a really cool setup, and we were evaluating those three groups. But what we did is we used this quick view camera to obtain images. It has a small magnification, a 15x magnification, and you can put on a filter with a blue light that can then image the fluorescence. And the images were then analyzed using image pro analysis, which allows an assessment of the green, the fluorescent green pixels in a defined region to allow us to quantify how much fluorescence was in a particular area. So the experimental method was applied to 57 dogs that were all previously evaluated to have normal baseline ocular health. And then we were able to obtain images at several time points from 0 application up to 180 minutes. And all of these were double masked, with the exception of Dottie, who was our sheepadoodle in our pilot study. So you can see we had a number of different breeds in each group, and relatively comparable ages in those groups. So we were successfully able to modify the HA and add the fluorescent tag, which allowed us to image the residence, the time of the two groups we were looking at. And we were able to identify the longest saline time is about 45 minutes. For linear HA, we were able to see it on some ocular surface up to about 120 minutes. And for cross-linked HA, we were able to image that in contact with ocular surfaces until about 180 minutes. So that's really an extended period of time. We did notice some really interesting things, including that the cross-linked HA exhibits this dual phase behavior, where in the phase 1 you'll see this initial kind of microgel coverage, and then following that you'll see a medial canthus accumulation, which is kind of the longer term time points. So these are some representative images from the study, and here you'll kind of see that initial coverage phase, and then later out here the medial canthus deposit phase. But this is cross-linked HA and these are all different animals, so you can see there's some variation between them. Then linear HA here, and then our saline control here. So saline really was all but gone by 45 minutes. We were able to see the linear HA in just a couple animals until 120 minutes, but we were able to see the cross-linked HA in some all the way out to here to 180 minutes. And this is just a little closer look at the microgel coverage that we see early on with the cross-linked HA, and you can see how it looks a little different than the linear HA. So now I want to just move on to talking more about how this cross-linked HA can support in healing in the corneal wounds, ulcers, and injuries. Our technology is also being developed by a human health company called Kyora, and this is a little excerpt from their corporate presentation, but they were able to show in a human clinical trial with about 400 patients that not only did they have really strong safety profiles, but that their efficacy profile was better than they expected. So they were able to improve product stability, they had longer retention on the ocular surface, and no blurring in vision, and really outperformed their contact bandage lens control, which was their gold standard of care in this study. So we wanted to take that study and then see if we could do a similar type study, but in target species. So we've been working closely with Dr. Fabiano Montiani, who I know is there with you today, and he has been doing some really beautiful work documenting the healing of the cornea with the application of our bioenhance technology. So he looked at a rat corneal defect model and did a healing time assessment. Here we are looking actually at our cross-linked HA versus amniotic eye drops, but really I'm just showing here representative images of the cross-linked HA and then of the control, which had no treatment. And he was able to see a statistically significant difference between the control and the treated, and an improvement in healing. When he looked at histopathology results, and you can see here, so here's a normal rat cornea on the saline, but some intermediate healing time points of the HA hydrogel, and then looking at the HA hydrogel at 72 hours, you can see really organized healing that is looking really close to what you would see in a normal uninjured cornea. We're also looking at doing a study here at the University of Georgia with Dr. Kate Myrna, who is part of our U.S. Scientific Advisory Board, doing a controlled masked study for the healing of scad ulcers. So this is an upcoming study we're hoping to get initiated here in the beginning of 2023, and so we're excited to see results coming from that to complement what Dr. Montiani has already performed. This is a poster that we just also presented at ACVO, again detailing some of this beautiful work here, but also talking about combination therapies. So this is this is kind of where we're headed with our bioenhanced technology, and hoping to see big movements here in the next year on combination products. So we've been working in-house to identify combinations of a number of different active pharmaceutical ingredients with our bioenhanced gel, and then really looking at how does that combination impact both the efficacy of the API and also the physical properties of the gel. So we've looked at a number of different antimicrobials. We've looked at afloxacin and a neopolybacitracin composition, and also with tobramycin. And so far, we've been able to see some really great results showing that, especially for these water-soluble compounds, that simple mixing, you're able to get a product that doesn't impact the physical properties of the hydrogel, but also is able to maintain or even improve the efficacy of the drug. So in these three examples here, we are looking at the zone of inhibition. So how effective was the antibiotic that we are looking at against these strains of bacteria, and we're able to determine that there was no impact from the combinations. So the APIs were all performing as they were in their controls. But interestingly enough, when we combined our hydrogel with ganciclovir as an antiviral in a topical type formulation, then in this in vitro study, we were able to see an increase in efficacy and potency, really, of the antiviral activities, even above the citalopravir control that we were using. So this was some really exciting data that we're hoping to pursue there too. And that's all I have today. And if you have any questions or want any further information, I'm always open to emails or meetings, and would love to answer any questions. And I wanted to thank DrogaVET for allowing us this opportunity to share our products with you. And we're excited to see the Brazilian market come into play. Thank you so much.